Einen wunderschönen Montagabend zusammen. Heute mit einem internationalen Gast. Hello, Randy.
Hello, Laker.
Randy Oliver is an American beekeeper, I think in California.
Yes.
Is this correct? Biologist by...
Training.
not by trade, but by training.
Yes.
And you use this training to improve not only your beekeeping, but the beekeeping of all your colleagues who work with you. And I'm very glad you do this. I already used a lot of your material you present on your webpage, Scientific Beekeeping, to improve my own streams here on the Montagsimker channel. And I'm very glad that you join us today with this special theme of selective breeding for mite resistance, because that's a topic with the Internationale Arbeitstagung in der Schweiz will have as background theme. And we are a little bit preparing this meeting with your talk here. As I'm not so used to speaking English, I can understand pretty fine. No problem to understand what you are saying, but the other way around is a little bit cumbersome. So I give you the word and your presentation.
Yeah. Ich spreche nur ein bisschen Deutsch.
Oh, that's very nice. So I could have started in German and you would have understood. Nice.
No.
So, selective breeding for mite resistance. I read about this for years, and I'm not sure how much our audience already knows about this. So I ask you to start with the beginning.
Okay. Well, let me, as an introduction, people have been breeding for many years and often making it very complicated with lots of work in the selection process. And what's happened in the last few years, there have been a few reviews that have found that most of the programs have not succeeded very well, and they've made them too complicated. And actually, the program that I've been using is probably the most successful way to do it. So I'm writing, actually I wrote this morning, I'm writing an article for the American Bee Journal. It will be on my website in a couple of months. I'm going step by step in a series of articles, how to set up a selective breeding program of your own. But I'll go through this one today. Go ahead, next. Okay, next. Okay, so in the integrated pest management pyramid for any kind of plant or animal that's managed by people, you normally start here down the bottom with resistant stock. So where I planted a bunch of grapevines, different types of grapevines at my place, some of them got mildew every year. So I just yanked them out and replaced them with cultivars that do not get mildew. That's the easiest way to solve the problem rather than having to spray them or scrub the leaves off. Same thing with honeybees. Ideally, we would have started with resistant stock. And then you go up to physical and mechanical methods, powdered sugar shake, screen bottoms, those kinds of things, heat treatment. And then you start monitoring your pest level. And we do that with mite washes for the Varroa mite. If all these things fail, then you go to biopesticides. That would be like the formic acid or thymol or oxalic acid. And as a last resort, you finally go to synthetic chemicals. Unfortunately, in most countries, we skipped all these steps and went straight to synthetic chemicals. And that removed the evolutionary pressure for the honeybee to evolve resistance to Varroa mite. So we've created our own problem. And now as our synthetic chemicals, mainly amitraz, are failing worldwide, beekeepers worldwide are suddenly in a position where the treatments they've used for a number of years are no longer working, and we're seeing really big problems. I've foreseen this, so we started back in 2001, we shifted to only using the natural miticides. We haven't used the synthetic since 2001. And in 2017, we started a very strong selective breeding program for Varroa resistance. Next. Okay, I'll go pretty fast, so keep your finger on the trigger. So I was in Azerbaijan last year, and speaking to them, they had some problems. And a Russian delegate said, hey, you have tropolelaps in Russia and probably in Azerbaijan now. Next. Now, you should be able to click that screen and get a video to play. Ledger.
No, I can't play this video, this program.
Okay. They showed a beekeeper from Azerbaijan emailed me after my visit and showed a dissection of these cells and 15 to 20 tropolelaps mite coming out of each cell. So it's knocking on your door. You guys are going to get tropolelaps very, very soon. Next. And we've spent 2 generations in the United States, even longer in Europe, fighting Varroa mite with fly swatters and Band-Aids, not looking for a permanent solution. And this is discouraging. Next. We're going to go through these really fast now. The only long-term solution to Varroa and tropolelaps will be selective breeding. Next. So the question is, why is this happening? Why are we waiting so long? Next. Next. So there's lots of queen producers in the United States, and some are queen breeders that actually select for breeding. Others are producers that buy bee stock from a breeder, and then they produce hundreds of thousands of queens. I don't know that happens so much over in your country, but it's not hard to produce queen bees. Next. But it is difficult to do selective breeding with honeybees. Next. So every aspect of honeybee reproductive biology is contrary to selective breeding. They want to diversify, keep their gene pool as diverse as possible, do recombinations, multiple fathers for the colony. So that makes selective breeding for the honeybee probably more difficult than for any other animal or plant on Earth. So it is a challenge, but it can be done. Next. So you have some options for how to breed for mite resistance. Next. You can just take the bond method named by John Kiefer in France, where you just put out a bunch of colonies, let most of them die, and then you breed from the survivors. The problem is most beekeepers would go out of business with that method, so it's not very popular with most of us. Next. Now you can do very complicated things. You can do like Arista does with the buckfast and do all kinds of brood dissection and progeny tracking and all these things. Rather complicated. Most beekeepers are not going to do this. Most queen producers will not do this. Next. And that brood dissection, it is informative, but doesn't necessarily tell us what we need to know about resistance. Next. Because it focuses on only one trait. In the US, it did a lot of this liquid nitrogen freeze-killed hygiene test. Well, this is very good to breed bees that are resistant to American foul brood, but not for Varroa mite. Next. You can do instrumental insemination, which may speed up the process, but we found is not necessary. The other thing is marker-assisted selection. That's genetic. So you analyze the DNA of the bees, and you look for markers that correlate with resistance. Well, there's like 70 different markers that correlate with resistance, and they've been unable to use that genetic genotyping in order to advance selective breeding. Next. And this is one of the reviews that came out recently that says, recent review revealed there's no clear evidence between any of those proxies that we use for mite resistance. Next. It's not like queen breeding is something new. These are 2 books I picked up recently over in London, one from 1889, one from 1924, about bee breeding and scientific queen rearing. There's nothing new about this. We can do this without recent scientific advances. Next. So back in the day, we simply just bred for workable stock that was not too spicy, that produced honey. And all we're going to do now is just add resistance to Varroa. Next. My suggestion is keep it simple. Don't make it more complicated than necessary. Next. So there's no magic in here. It's just a simple process of selection at each generation. Next. You have two kinds of selection, negative selection and positive selection. Next. So negative selection, don't breed from bees that sting a whole lot, or don't breed from bees that don't make any honey. That's negative selection. So you eliminate those queens. Positive selection is, go ahead, is where you take a plant like this is mustard plant right here. If you breed for plants that make larger flower heads, you wind up with cauliflower and broccoli. This is the same species of plant right here, just selectively bred. Or you can breed for larger leaf clusters, and then you get cabbage. Or you can breed for larger buds on the stalk, and that's Brussels sprouts. These are all cultivars of this species of plant that have been selectively bred by humans before the word genetics was ever invented or before the word science was ever invented. You don't have to be a scientist to do the selective breeding. Next. So back in 1999, John Harbaugh and Jeff Harris from the United States Department of Agriculture, they said it all. Compare the growth of mite populations in each colony and determine which bees are more resistant. All you got to measure is the number of mites in the colony, the mite infestation rate. Next. If you do any other metric, you're telling the bees how to do the job. And they may figure out ways to do it that you would never have thought of. So to me, to use a proxy for resistance, such as VSH or Harbaugh selection, you're missing the point. Let the evolution figure out how to do the job, and you just reward it if they get the job done. Next. So here's what we do. We started a very intense selective breeding program back in 2017. For the first time, we mite washed every one of the colonies in our operation. We were running about 1,500 hives at that time. So we went out and took mite washes from every single one. And one of those colonies had a mite count of 0. We called her Queen Zero. So we did not treat that colony. And then we went back a month later, and she still had a count of 0. We went back over the next several months, and that mite count never got above 0. So we said, wow, there's no magic that has to happen. We already have the genes for resistance in our own breeding population. All that we need to do is just select for them. We don't need to bring in any outside stock. We don't have to wait for a mutation. The genetics are already there. Next. And I wrote about this. It's on my website, telling me how to do this. If you had 1,000 hives, it would take about 100 hours of manpower every year to do this program. So not a whole lot. And if you Germans got together and collaborated or with a project like this, it's not that much work. Next. And we use only traditional breeding, nothing fancy, no fancy equipment at all. Next. You can start with your own locally adapted stock. You could possibly since honeybee is native in your area, there might be a locally adapted free living stock, or you can start with a line already selectively bred for resistance. Next. But main thing is you don't have to create anything new. The alleles for resistance are already out there in our population. You just got to put them together. Next. So this is the only thing we do. Our only measurement we take is mite washes and count how many mites. This colony would never make it into the breeding program because you can see there's 2 mites there. That's too many for us. We only want 0 or 1 mite all throughout the year. And there are plenty of colonies in our operation that do that all by themselves. Next. Okay, so first you have to do is wait till the mites build up. Sometime in the summertime, whenever your mite count, you can't do selective variation until the average mite count is around 5 or 10 in your colonies. Then at that point, you can tell the ones that are high are out of the program, and the ones that are still zeros at that time would be the ones you breed from. But you can't do that until the mite count overall has built up a little bit. Next. So what we do is every spring, we requeen all our hives. We split them into nukes and requeen them. Next. Then we produce enough queen cells to requeen every one of them, only from queens of colonies that needed no mite treatment at all for the entire last year and kept their mite counts down to 0. So we only breed every year from colonies that needed no treatments for the whole year and kept mite counts down to 0. Next. And then we dribble each nuke with oxalic acid on day 18 after we start the nuke. That knocks out most of the mites. So all these nukes now start out with relatively low mite counts with a new queen from a mother of a mite-resistant colony. And we breed off of about 30 different queen mothers every year to maintain genetic diversity. That's very important. Next. So we sell 1,000 nukes and we make another 2,500 or so for ourselves, spread them out in our yards. Every one of those hives started with a virgin queen from a mother of a mite-resistant colony. Now, when I say that, I don't say with a mite-resistant queen. There's no such thing as a mite-resistant queen. The queens don't affect the mites directly at all. It's her offspring that take care of it. So all I can say is the mother of a mite-resistant colony. And we don't know how much of those genetics came from the mother and how much came from the drones that she mated with. Next. And this is our home yard right here. And every one of these X's is an out yard, one of our other out yards, every one of them with 20 to 40 colonies in them. So we flood our environment with our own drones and we can control our matings that way. Next. Then we also give free queen cells to all of our neighbors every year. So they will carry our genetics. So their drones from their colonies will not interfere with our breeding program. All the beekeepers in my county are happy to use our stock because that allows us all to work towards developing mite resistance in our county. Next. And then we start the Varroa race. Those queens are mated. And now we say, Varroa, ready, set, go, start reproducing. And we say, bees, stop them, keep them from reproducing. Next. Okay, then we have to wait until those colonies build up to strong, usually in sometime, started out in early June. Now we usually have to wait until August or September until the mite counts get high enough for us to selectively select for or identify colonies that have maintained 0 mite counts. Next. So this is when we started our breeding program. We'd split our colonies here in the middle of March, and this is my mite model. It shows the total population of bees right here. Shows the amount of sealed breed in the colony, shows the Varroa population, and shows what your mite wash count would be. So back then, by early June, the average mite wash count in our colonies would be like 5 or 6, with some colonies up to counts of 20, even 30 mites. But at this point, if you find one with a 0, you know she's probably resistant. So what we do at that point, we mark those colonies that have 0 counts on a tag with the date and the mite count, and we call those potential breeders. They're not breeders yet, but they might be breeders. Everybody else we treat. We give them oxalic acid or formic. No colonies die, okay? But we don't treat the ones that are potential breeders. We leave them right in the yard. They get no special treatment. And we wait a month, and then we wash them again. If the mite count went up, we treat them and put an X through their tag. And we did again for a 3rd month, for a 4th month. Finally, in November, when the mite counts should be the highest, they still got to be 0s or they don't make grade. Then we let them go over the winter. And even the mite count may go up a little bit when they shut down brood rearing and all the mites come out of the brood. But if they need to take it back down to 0 by themselves to make breeder grade. Next. So no colonies die in the process because we treat anybody that has that has even low mite counts all get treated. Next. But the trick is we use mechanical agitators that I've designed and built, and now I have a friend who is building them and selling them worldwide to allow groups or large beekeepers to do very, very fast mite washes. So a team of 4 of us we can do a mite wash a minute, average a mite wash a minute. So a yard of 40 hives in 40 minutes, we know the mite count of every hive in that yard. It's not unusual for us to do 300 mite washes in an afternoon. So that by doing this with these agitators, we can really move quickly. Next. We have also invented a very inexpensive and efficient mite wash cup for hand washing or with the agitator. And my friend Jake, standing here, I don't sell anything myself. I don't promote anything. If I come up with a good invention, I give it away to a friend and say, "Go ahead, start selling it." So he is selling these. He is not shipping to Europe yet. He's shipping down to Australia and New Zealand. But these are much, much handier than the ones that are on the shelf. These guys copied my design years ago, sent me prototypes, and then made what I think is a terrible mite wash cup. This one works way better. Next. And then I've designed these mechanical agitators. They're battery powered. You take them out in the field, you put the cup on there, you push a button, and it automatically agitates for X number of seconds, whatever you set it to. And very, very quick mite washing. Has a built-in magnifying mirror to very easily identify the mites. So we make these for ourselves, and Jake is also now selling these. This allows a individual large beekeeper or a club or organization to do very rapid mite wash counts. Next. Yeah, two of us, here's my assistant Rose, two of us together. I can't read this because there's something in front of it, but we can do the, let's see, can I hide? I can't hide it. My screen's blocked there.
You can do 35 hives an hour.
Yeah, so the two of us can do very, very fast mite wash counts. Next. Okay, first thing, people say, oh, well, resistant bees have to be aggressive. That's absolutely untrue. Next. So in California, it's hot. This is how I dress when I'm doing mite washes all afternoon. Okay, I may be doing 50 or 70 mite washes on it. Don't get stung. So we keep very, very gentle bees in our operation because it's hot in California, and this is how we dress when we're working bees during the summer. So that's a fallacy that resistant bees have to be aggressive. Next. Also a fallacy that they won't make honey. We only breed from colonies that are above yard average honey production, and actually the feedback we get is that our bees make more honey than any other bees that people buy. So you can have mite resistance and not sacrifice anything. You don't give up gentleness. You don't give up honey production. Next. Yeah, the zero count colonies often make the most honey in the yard. Next. And here's for example from some of our breeders of mite wash counts over the course of the year. And they're all at zero a year later, all by themselves, with no treatments whatsoever. Now what we do, if we open one up during the season and they make more honey than all the rest of the hives in the yard, we put a star on the label right up here. Or if they're exceptionally gentle, we'll put a star on there. If there's something else we really like about that colony, we'll put a star. So not only are they mite resistant, look at that one. We never found a single mite in 5 mite washes over the course of the year. And she got 2 stars. So one at each different time point saying this is the best hive in the yard. So these are the ones that we like to breed from. Next. And next spring, we graft off of those queens, rear a bunch of queen cells from each one of those, again, about 30 daughter queens, or queen mothers, to maintain genetic diversity and start the process all over again. Next. Now, one of the mistakes that many breeding programs do is they think that the grass is greener on the other side of the fence and that maybe they should bring new stock in after you start. If you're doing a selective breeding program, you don't switch horses in the middle of the race. Okay, you stick with what you got. Don't bring in any outside stock after you start. Next. And again, as I said, there's no such thing as a resistant queen. It's all about who that queen mated with. Next. What you got to realize is the genetics of the drones do not reflect the genetics of the whole pool of their parents, but of their grandparents. So there's a 2-year lag in the genetics of the drone pool. So if you're doing selective breeding, next. So right here, this is our progress from 2017 to, I made this graph before 2025. This is the proportion of colonies in our operations that are completely resistant to mites. We're up to about a third of our colonies need no mite treatments whatsoever. The blues are the ones that maybe need 1 treatment a year. But right here, if we took a queen from a resistant colony, she'd be mating with a drone pool, of which 95% of the drones came from non-resistant colonies. So that's why the progress is fairly slow, because there's a 2-year lag always in the matings, because the genetics of the drones reflect the genetics of their grandparents, which is really good evolutionarily wise for the honeybee, because if a forest fire or storm wipes out all the colonies, the few colonies remaining can carry the genetic diversity of the whole population rather than just the survivors. But for a breeding program, that puts a 2-year lag on it in every step. Next. So this is typical from a 24 buyer, but they said that about a third of the colonies were completely resistant, half about moderately, and a very 23% needed more than 1 treatment. So this is pretty typical, and we're doing even better this year. Eric, my son, went out and mite washed 250 colonies, and only 5 of them out of 250 had high mite levels in the 10s or 20s. Another 5 had mite counts in the 3s and 4s. And out of that 250, roughly 240 colonies still were at 0 or 1 mite counts this late in the season with no treatment. So it's looking pretty good. Oh, next. And the point is, even a partially resistant colony may only require 1 treatment a year with an organic treatment. So it just makes mite management much easier. You don't have to get to the point of fully resistant right away. By the time you have most of your colonies partially resistant, mite management becomes much easier. Varroa is not a major problem in our operation anymore. My main problem right now, as a researcher, I can't find enough colonies with mites in them to test miticides for efficacy. I tell my sons, well, a colony with mites is gold for me. Don't treat it, bring it home. I need colonies with mites so I can test different treatments. So it's a good problem to have. Next. Okay, so someday Varroa is only going to be considered to be a minor problem. Same thing in South Africa, where they never even talk about Varroa, even though all the hives have a low level. And that, if we ever get on this bandwagon of selection for resistance, we could all be in this point. Now, I cut this PowerPoint off here because I have more slides on how the bees actually do fight the mite, but I didn't know if you wanted to do question and answers or talk at this point, or go on to the other subject.
There is one question I can use. Doesn't it make sense to breed from colonies in June to double the speed to achieve the goal of resistance instead of waiting until the next season? He is thinking about 2 breeding steps in a year.
I understand. Yeah, that's definitely a possibility. We really want to give those colonies a long chance to prove themselves. So theoretically, he's absolutely right. You may be able to increase it to double the rate.
That's the only question for this part. Perhaps we continue. Perhaps some questions will arise later. I hope so. I just had a question on my own, but didn't write it down. Sorry.
Yeah, if you had a question,
yes, one question I had. You started with the problem of Tropilelaps mites. Do you expect that the same mechanisms that work for Varroa will work for Tropilelaps?
That's an excellent question. The Apis cerana, the Asian honeybee, the native host of Varroa, is not. In fact, it is resistant to Tropilelaps also. Tropilelaps came from Apis dorsata, the giant honeybee, but Apis cerana is resistant to it. My suspicion is that the same mechanisms will work with my bees. I'm trying to figure out a way of getting some of my stock shipped over to Asia to test it. So that's a question that's very much on the top of my mind. That I think this will probably be a big head start on Tropilelaps. We don't have it yet in the United States, and we may be able to keep it out, but you guys will not be able to keep it out. It is going to hit you guys pretty soon. So I would be selective breeding for Varroa resistance really, really hard right now.
Can't hurt against Tropilelaps. I don't think so, yes. Okay. A question you already answered. Do you do artificial insemination in your breeding program?
We don't, because we're doing a demonstration project. I started this out as a demonstration project for the other queen producers. So I'm not trying to do it the fastest way possible. I'm doing it on a way that any beekeeper can do who doesn't have equipment. So yes, I could probably accelerate the progress with instrumental insemination, but I'm deliberately avoiding anything fancy. How's that for an answer?
Oh, that's fine with me. I tend to argue in a similar direction, especially if you want to use breeding in a bigger group of beekeepers. You can't use high-tech approaches, right. And you need something that everybody can do.
Yes.
One question, in order to succeed with this strategy, don't you need local dominance from your drone population?
Absolutely. So what we do is, as I showed in my picture, we flood our own mating yards. We come back from almond pollination with our colonies completely chock full of our drones, and we just flood the local area. The other thing we do, we've got a collaboration with a major queen producer who is a friend of mine, and he wanted to produce some of our stock. And we said, oh no, you wouldn't be able to control the drones. He said, well, actually, I lease a 25,000 acre, so that would be 12,000 hectare ranch, cattle ranch. And I have only rights to put honeybee hives on there. So he cleared all the hives off. We moved in 450 of our colonies for drone mothers, and he's mass producing tens of thousands of our queens. And the return from the buyers from the last year, he's ramping up to try to do 100,000 of them this year. The people have liked them so much, and they're liking them not even necessarily for the mite resistance. They're liking them because they make so much honey and they are so gentle. And mite resistance is a bonus to them.
And it will be a bigger bonus if they are resistant against Tropilelaps.
Oh, yeah.
One question, you already answered this also. So you would never introduce other genetics, but always build on the existing ones on your own.
Otherwise, it's not a selective breeding program. It's gambling. It's like if you bring in new genetics, what you can do is you can totally ruin the mite resistance. Takes a system of finely tuned interaction between a number of genes and regulatory cascades. So both behavioral and coding for proteins. It's like taking a Ferrari and a Porsche and a Mercedes and mixing up all the parts and putting them together to make a car. You're not going to get a good car. You're going to mess up any one of those finely tuned systems that go together. So when you bring in new genetics, you have more chance of screwing things up than you have of improving it.
Here's a question regarding your mite washes, your mite counts. Would the count with sticky boards, with the screen board, work also as a measurement for mite?
Sure. Yeah.
Levels?
If you saw virtually no mites on the sticky board, I'm not sure that would work. It should work because half the mice on the sticky board generally come out of emerging brood, that those mice that aren't quite old enough to mature. But if you're not seeing any mites build up on the sticky board, that should be another reasonable assessment. Not that it's going to take, it actually takes longer to do a sticky board count than it does to do a mite wash.
If you're precise, for sure.
If you have a system. Yep.
How do you choose your drone colonies?
The drone colonies are those that were potential breeders that didn't quite make breeder grade. So they have maybe slightly higher mite counts, not quite as good as the breeders, but pretty good. We make sure we don't take any that had any kind of high mite count and required major treatment. Keep those away from the mating yards.
Do you have a ratio with how many hive or queens from the last year you keep for this purpose, and how many you requeen with your queens from the current year?
We requeen everybody. We run our queens for a year and a half. So they make a bunch of nukes in the spring, but we don't kill the old queen. If she looks good, we put her in a nuke and let her build up also. And then in August, when normally our queens get superseded after a year and a half, the bees normally replace them because they run out of sperm. At that point, we use those bees to strengthen first year colonies that haven't built up yet. So then on our breeders, we do run those for 2 to 3 years just to maintain their genetics and to do progeny tracking, which means it's not so much what the queen's colony does, it's what her daughter colonies do. So we just started doing that. So on every tag now, we're writing down the mother of that colony. So essentially, it's her granddaughters that are actually fighting the mites. Then we want to check what the great-granddaughters are doing. And if the great-granddaughter is doing it, that means it's probably in the maternal line of genetics and did not come from a drone. So that's what we're looking for now, tracking it's called progeny tracking. So you track who the mother colony was, but that means that queen has to live for 3 years.
Well, I believe I have an answer to this, because I use instrumental insemination and know my mothers and drone colonies for generations.
Right.
And I'd say it's 50-50. 50% of you need a mother of a resistant hive as your mother as your breeder, and you need a fully resistant colony. Okay, you need 1 generation more because of the drone effect to achieve a high probability for a resistant. So the question I have, because I haven't done single drone inseminations, is what proportion of the drones need to carry those recessive genes? Does it just take, if the queen mates with 20 drones, with 2 drones with resistant genes, be enough to have enough workers in the colony to exhibit those resistant traits? I can't tell you this from now because my parental side is too strictly controlled. It's too small, and the queens are too heavily controlled. So I have probably too high a quote of resistant bees in the colony to tell you this a lower bound for this effect to take to be have result in this effect.
It'd be very interesting to find out.
Indeed, yes. Perhaps okay. Does the supply of good pollen and pollen substitutes or hive alive or something like this have an effect on mite resistance?
Not that I've noticed in any way. Yeah, mite resistance is behavior of the bees. Not only behaviors, it's also some of the things like their pheromonal production. So one of the ways that they fight the mite is by the fifth instar larvae delaying the time when they shift the pheromonal signals to the nurse bees, and that screws up mite reproduction. So that's not behavioral, that's actually physiological.
Mm-hmm. Do you have a list on your website that would tell us which traits come from the queen and which from the drones? Or do you have knowledge about this?
There are questions. What you're talking about is maternal and paternal effects. The genes, it's the same genes coming from both of them. It's the same allele. All the bees have the same genes. They have different alleles of each gene. There's not a difference in the genes. There's a difference in the alleles of each gene. And whether those alleles are expressed differently, if they come from the drone or the mother, we're not really clear on. So it's a possibility, but have not investigated that.
Okay. Perhaps we should start with the second part?
Sure.
Or do you want to finish it off, this discussion?
It's up to you. We can go on for
how much time do you expect to need for this?
I don't know, maybe 10 minutes.
Okay, then let's continue.
Okay, next. Okay, so one of the things that's bred for sometimes is the biting behavior, and that could help, but from what I've seen, that's not the major thing. When I model Varroa buildup, if the normal attrition rate is about 0.5% a day of the mite population die of natural causes. If on my model I increase that up to 2%, the mice just disappear from the hive. So if biting really did it, we would see the mite biters disappear the mice, but that doesn't happen. So it may help, but it's not the major thing. Next. The Achilles heel of Varroa is its rate of reproductive success. How many offspring the female has per reproductive cycle. Next. So you don't have to kill a single mite to completely control Varroa. That's a really important point. You don't have to kill a mite. All you got to do is cut their rate of reproduction in half, and they will disappear from the hive. Next. So I ran 2 simulations here. The normal value on a hive for, let's see, do I have this? Okay. The normal reproductive rate, this is called the R value, the instantaneous rate of reproduction. And that's what the increase that you'd have. Actually, that's not, I'm sorry, it's not the R value. This is the average number of mated female offspring that a female mite, a foundress mite, has in a reproductive cycle. A little bit less than 1.5 on average come out mated and ready to go. And that would be our normal growth curve up here showing how fast they go. If I cut this down to just 1/2 of that, look at the growth curve. So you cut the rate of reproduction in half. You didn't kill a single mite, but the mites cannot build up in the colony. So that's the key thing here is suppressing their reproduction. You don't have to kill a mite, just mess up the reproduction. Next.
One question at this point. Do we know anything about reproduction rates of Tropilelaps?
Yes, we do. Much, much faster than Varroa.
That's the problem I see.
Yeah, much faster. So one of the traits that we see a lot is this bald brood, this uncapping. And what that does, the female mite, when she gets uncapped, often leaves, but she doesn't get killed. And maybe the offspring dry out, but mainly you have messed up her. Normally a female mite in her lifetime does about 2.5 reproductive cycles. When she leaves this, her timing is all off on her ovulation and the pheromonal signals. So she may miss out on one of the reproductive cycles. So simply by uncapping a cell and then recapping it over after the female is left may cut the rate of reproduction in about half for that female mite. Next. But that alone won't do it. And what I find is you can put a sticky bikini wax pad that women use, or maybe you use it, I don't know, over the top of the cappings and rip off the cappings. You can see the silk capping right here. The dark ones have been uncapped and recapped. So even in my non-resistant colonies, I see a fairly high level of recapping behavior. So you need something else just besides just the uncapping and recapping. Next. So let's skip that one. What you need is Varroa-sensitive hygiene then. So not only do they just uncap and recap, but in some of them, they will actually chew that mite out of there and make sure that they kill all the offspring too. So it appears to be a combination of the uncapping behavior and some Varroa-sensitive hygiene. Now, those are things that we see in our resistant ones. We have not tested to see if there are other traits, but these two are very, very obvious. Next. And we see a combination of them. So here's uncapping behavior, and there's Varroa-sensitive hygiene happening in the same hive. Next. So in the first round of brood in the springtime, they usually look like this. Very, very nice, solid patterns. Next. But by August, in a resistant colony, the patterns look like this. So that's telling us, yes, there's still mites in those hives, but they're doing very strong Varroa-sensitive hygiene and keeping them from reproducing. So this hive, we know there's mites in here, but we get a mite count of 0 if we do a mite wash. Next. The other one is the suppression of mite reproduction, where the larvae and the pupae fight back. So what they can do is, by changing the pheromones they put out, they can delay the egg-laying initiation rate of the female mite. So this is like molecular warfare between the bees and the mite. Then out of Norway, they're doing a different one where some of the bees down-regulate the expression of certain genes that make ecdysteroids, because the mites use those sterols directly from the bee in order to produce their eggshells. And they can't make it themselves. They have to get them from the bee. So the bees, if they down-regulate expression of those, they can prevent the mites from being able to lay eggs. Now, who would have ever thought of breeding for that? So
I wouldn't know to test how to breed. You need to be able to test this to breed for this.
So exactly, why test anything? Just test to see whether they have mite increase or not. There's no reason that you have to understand how they do it in order to breed for resistance. That's my whole point. It's not important to understand how they do it. Just breed. Tell them the job description. The job description is don't let mites build up. If they meet the job description, promote them, breed off of them. If they don't, do not breed off of them. Very, very easy. Next.
Especially the second part. Don't breed.
Yes. Yeah, I think we're going to flip. You can flip to them a little fast, and I'll tell you if I want to pause on one. Let's see. Yeah, so this is, so these are, again, the pupae and larvae. It's pitch black inside the hive. Nobody sees anything. It's all done by smell. So the larvae and pupae communicate with the nurse bees and also inadvertently with the mites by the pheromones and the odors that they put off their bodies. So if they switch those, they can screw up mite reproduction. Next. And they put out a sacrifice me pheromone. And this is what Apis cerana does, that the larvae put out a sacrifice me pheromone if they get any mite saliva injected into their bodies. So that may be what Apis mellifera is starting to do. The other thing is that we see, this is very frequent. Mite count of 2 in July, drop down on itself in August to a 1, and by November down to a 0. So we see this all the time, that the colonies are able to bring their mite counts down by themselves. Next. And we found that out once we went in a controlled trial. I put in a bunch of colonies that were failed potential breeders, that the mite counts are too high. So I thought, well, they're not resistant. Use them for the control group. And when the mite counts went up, the colonies go, oh, holy shit, the mite counts are too high. And they did this, took down the mite counts by themselves with no treatment at all. We see this all the time with our resistant colonies. So they may not be good enough to make breeder grade, but the mites come up, they jump into action. Next, I'll show you a picture of it. So here's a picture of them. Once that mite count got up to 15, that activated something, and they just tore open the brood and removed all the mites from the brood. So that's typical with these guys.
We call this panic VSH.
You call it what?
Panic VSH.
Oh yeah, yeah, very, very much. And we see that there's something kicks in with them. Next. Yeah, so there's all kinds of things that we don't know, but we don't need to know. We can go ahead and breed for mite-resistant bees without knowing exactly how they do it. It's fascinating to understand why, but it's not necessary for the reproductive process. I think that's probably good on this if there's more questions.
Okay, let's check. That's basically a thank you for your presentation. Sehr interessant. Very interesting. Somebody's asking, why didn't you start 10 years ago?
Well, I did start, but I was fooling myself. You really have to get serious about this. You can kid yourself thinking you're doing a selective breeding program, but unless you control the drone pool and really keep track of people, you're really kidding yourself. And I did. And many beekeepers kid themselves for a number of years. And that's why I'm publishing this, saying, hey, stop kidding yourself. Here's how to do it, make it work.
This is an interesting one. If I manually uncap the brood several times, this would have an effect without the use of medication. Mm-hmm. This seems feasible to me and ethically preferable to total brood removal.
You must be retired.
And actually, it's not improving the breeding process. That's just a way of treating the bees.
Treating. Yep.
Sehr guter Vortrag. Very nice presentation. Thank you.
Thank you.
This is interesting. Do you allow visitors to come over for 10 days, 2 weeks or something to join the process and learn?
We have visitors come by. We don't have housing for them, but I often have people stop by and see. And I'm happy to share that information with anybody. We're going to be doing more filming of it and videos so people can see it. I've had a number of commercial beekeepers and queen producers stop by being very skeptical. And I say, well, let's just go out and do some mite washes and see. And they go, wow, were we wrong? They're beautiful bees and completely resistant. So, yeah, we're not trying to sell a product. I'm just trying to help beekeepers worldwide figure out how to do it.
So how big is your breeding population?
It's about 1,500 to 2,000 hives.
Okay. Do you have a guess how small such a breeding population could become to be viable and have some results?
My guess, it would have to, like on Santa Cruz Island in California, there was a population of bees there, maybe about 100 colonies over the island. They intentionally introduced Varroa to try to control it because it was an invasive species, and that exterminated the bee population.
Okay.
So too small a population, you may not have enough genes, alleles already present. So it depends on that. If you have other beekeepers around with influencing your drone pool, that's going to stop you. So if you live on an island, you can get by with a much smaller population because you have no other outside drones coming in. If you have a bunch of outside drones coming in, then you're going to need several hundred colonies minimum to flood your drone pool.
That's a problem here.
Right.
Controlled.
So you have to try to get beekeepers to work together.
That's probably the only way I can think of, but that's even more difficult than keeping bees.
Yes, it is.
We call it like beekeepers is almost impossible.
Right. It's like herding cats. It's just almost impossible.
Do you have buckfast bees?
There are some buckfast in the United States, but I'm not sure how true bred they are. I've worked with buckfast bees when I visited Europe, and when I visited Germany. Yeah, I've seen buckfast there, and it looks like great bees. Yeah, use I live in a Mediterranean type climate, so we use Italian type bees as our starting population. So you're starting with a bee that's adapted to your climate, and breed for resistance from them.
So you would say it's independent of the breed, as long as it is locally adapted to your climate.
I can't guarantee that. There are certain alleles that have to be present. So whether your population already has those alleles, I don't know. It just so happened I found out those alleles were present in my population to at least do some of those mechanisms. I don't know that they're doing all the mechanisms possible. So you might have 5 different breeding programs in different areas, and they may all be successful, and they may fight mites in different ways. It's just like if you have one army that has swords and one army that has rifles and one army uses machetes, and one army uses poison, they might all win the battle, but they won the battle by different methods.
Okay. That's especially, you just give the job description, not the...
Exactly.
Okay. This seems to be the finish of the questions.
Okay, well, that's good timing. It's about time for lunch over here.
Just some words of appreciation. One question.
My motivation is the feedback that I get from beekeepers worldwide. The number of thank yous that I get every day. I'm retired now. I don't need to make money. I don't need any fame. I got no quest for that. But I just feel good because everywhere I go, the beekeepers thank me for what I do. And that's all the motivation I need.
Do you think that the alleles we need are rare?
Well, they would be less rare if you've had Varroa for a while. So you have a feral population, or a native population of bees out there living. If they're still
sure
living,
that we have, but yes.
My guess is what we've seen in the United States, even though they're not native to the United States, we have populations that, if you look at the mitochondrial DNA, they have been continuous germ lines without ever losing a female population. So they are independent of our managed bees all throughout the United States. So I would imagine in Europe, you do have populations of bees out there that are survivors that have contributed alleles to your managed population. So my guess is you would have alleles there, especially if you went out anywhere remote from where people are keeping bees and start looking at the colonies living in trees that are still alive after what? You've had Varroa for what? 60 years or something like that. If you still got populations alive out there, you have some alleles for resistance.
For sure. Okay. The one important question for the typical German beekeeper who has 10 to 15 or even less hives Is there a way to have resistant bees with only, let's say, those 10 hives?
The best thing you can do is support a breeding program. So either in the United States, what I'm pushing for is that most beekeepers in the United States, unlike Germany, buy their queen bees from a queen producer. And supply follows demand. When the demand by the consumer says, hey, we want resistant bees, the producers will listen to that. So in Germany, if you're making your own stock, there's not much chance unless you collaborate with a bunch of neighbors. And that's frustrating. I wish I could tell you otherwise, but it wouldn't be truthful to say you really can't do much if they're mating out with drones from other stocks.
I agree, but it's nice to hear confirmation for this.
Yeah.
Okay, thank you very much for your work.
You bet.
I'm very glad to have you here.
Mm-hmm.
And
it's nighttime there, right?
It's 9 p.m.
Okay, then good night.
I want to do one other thing. I hope it's working. I want to show you your web page.
Oh, yeah.
No, that's the wrong program.
If you go to the last slide in the presentation, I believe my web page is there.
No, there
should be a picture of it. No, okay.
Now I'm lost. Okay.
Yeah, scientificbeekeeping.com.
Scientific beekeeping. It's the page.
Yeah, and we're going to completely. I just hired Rose this winter. We hope to completely turn it around and make it much more user friendly.
Okay, the system just stops working. I was too quick changing buttons, so it's telling us we have to leave. You have to eat. We have good night.
Right. All right.
And thank you. Next week, I'm not sure what we will do next week. Perhaps I will do a short summary of your presentation in German for those who don't understand enough English to follow your presentation. Perhaps Ralf Sester, who is the second Monday beekeeper, will do something to tell about the current daily work at the beehives. And in 2 weeks, Dr. Ralf Büchler will join us and will do a presentation on non-chemical treatments, brood removal, queen caging, and those. And at the latest point, the week after this, I will do a translation of your presentation. So everybody who didn't understand what Randy was presenting, I'll do this. I tried to repeat what you did in German. I'm not sure, but I find it important that the content you brought to us is understood by everybody.
Wonderful. All right.
See you.
See you later. Bye.
Okay, stream is closed.
Okay.
We are on our own.